expertise de l'aspect cost effectiveness de ça, mais on a commencé à l'évaluer, y compris avec Jean-Paul Moati, au moins sur une situation en France, pas au Sénégal pour l'instant, mais en France, qui est celle des méningites, et on a montré que cette façon de faire du diagnostic rapide des méningites est effectivement cost-effective. Pour faire simple, le surcoût entraîné par le dispositif, les tests et ainsi de suite est largement contrebalancé par l'efficacité de la prise de décision médicale éclairée par le résultat des tests. En clair, dans le texte, la décision d'hospitaliser ou pas, vous en doutez, fait immédiatement basculer la balance financière du bon côté. Si vous êtes capable, en faisant du diagnostic rapide, d'éviter des hospitalisations indues, on peut tous comprendre que rapidement, le système est cost-effectif. On l'a montré au moins dans une situation, ça ne sera peut-être pas vrai pour toutes les situations, mais au moins sur la situation des méningites en France, c'est quelque chose qui est efficace. Donc pour conclure, une nouvelle façon, si vous voulez, de pratiquer notre métier de diagnostic des maladies infectieuses, de microbiologie, de façon rapide, encore une fois, d'une façon qui doit nécessairement éclairer la prise de décision médicale. Ça, c'est un point très très important. Deuxièmement, ceci est rendu possible par la technologie uniquement. Troisièmement, cette façon de faire que l'on a expérimentée au départ dans le périphérie de notre laboratoire cœur à Marseille est maintenant exportée et de façon, finalement je trouve comme vous, je bute comme vous sur l'adjectif sustainable, mais peut-être de façon pérenne, disons, et pérennisée maintenant sur un village. Comme l'indiquait Madame Touvenot, pour nous, c'est une étape zéro, c'est une preuve de concept, on est bien d'accord. Donc nous pensons avoir apporté la preuve du concept que l'on peut faire comme ça dans ce que j'appellerais les déserts médicaux. Ils existaient à Dielmo, au fin fond du Sénégal. En réalité, il y en a aussi en Europe et pas si loin que ça en France. Donc la capacité à implanter des petits laboratoires comme ça, grâce à la technologie, grâce aux outils de communication, grâce à une formation extrêmement rapide de personnel éventuellement n'ayant pas de formation spécifique au départ, et donc la possibilité de guider les médecins dans leur choix rationnel avec probablement, donc encore une fois, au moins sur certaines situations, du cost effectiveness. Je vous remercie de votre attention. Puis je répondrai aux questions en fin de session.
Merci beaucoup pour ce bel exemple aussi de transfert de technologie. Alors, vous ne nous avez pas dit si le cargo va de Marseille à Dakar, et ça serait là. Non. Donc, tant pis, mais c'est quand même bien intéressant. Donc, on va enchaîner sans plus tarder, comme je disais, par l'intervention de Mathieu Albert, de l'Institut Pasteur et de l'INSERM, de l'unité 818 de l'INSERM.
Thank you for the introduction, and I'd like to thank Jean-Paul Moati for the invitation to this conference. It's been a very interesting day so far, and quite strange for me as an immunologist of 15 years to find myself at a public health symposium, learning quite a bit and hopefully having something to share. So I'd like to speak for the next 20 or so minutes about some of our experiences in the context of work on hepatitis C viral infection, and specifically on what role I think point-of-care devices can play in terms of predicting response to treatment. And this work has been incredible collaboration, multiple different consortia that I'll try and highlight along the way. But just to say ahead of time that it's only through incredible collaborations with the ANRS site in Egypt run by Arnaud Fontenay and collaborations with Stanislas Paul at Hôpital Cochin that this has been possible. So I've been learning a lot about the creative destruction of medicine over the past couple of years. And I'm halfway through this book, and I recommend it to anyone who's sort of considering questions of personalized medicine. We already heard quite a bit this morning, I think some sobering comments, but I'm very excited about the technologies. We've already heard a couple of talks, notably from the keynote speaker, about how mobile phones have revolutionized communication systems. And I really believe that Eric Topol has got it correct, that we are going to witness an incredible revolution over the next 10 years as devices like this also revolutionize our medical care. And it's actually exactly in this line that we've built a new project around implementing point-of-care devices on iApps that could be read out on mobile communication. I'll come to this at the very end. So just to give you a little bit of background, my laboratory has been working on questions concerning the pathogenesis of hepatitis C infection. This is a worldwide health problem concerning between 150 and 170 million people worldwide. It is a flavivirus. It is actually the only flavivirus that is not vector transmitted. It is just through parental transmission that one becomes infected. It's a small RNA positive strand virus, and you can see here the representation of worldwide infections, and I'll come back to that in just a second. Just to review a couple of definitions, we've already spoken a lot about stratification of patients and predictive markers. I just want to highlight what I mean by personalized biomarker, which is really any biologic measurement that can be used to support patient group targeted therapies. I'm not sure if we mean the same thing in terms of N=1 clinical trials necessarily, but basically to take the strategies that are available and to adapt them to the individual person based on their, whether it be genetic or protein biomarker or general health measurements. And this really demands therapeutic need and implies innovative medicines and testing approaches. And I think, as I'll highlight, hepatitis C has really been an excellent example for us over the past decade. We're talking about trying to identify the right patient, the correct disease, the correct treatment, the right time for treatment, the optimal dose, the optimal response, and something that I'm more and more aware of, and for that reason very appreciative of the opportunity to speak today, it must come at an appropriate price. And really to that point, I want to highlight something that is happening within the viral hepatology, viral hepatitis community, which is the introduction of new and very exciting drugs. Over the past 2 decades, there's been incredible innovation. When I began my work on HCV, we were less than 50% response rates, quite ideal for questions of stratified biomarker research. Recently, with the introduction of Telaprevir and Boceprevir in 2011, we're now up to 70% response rates. And with all of the new drugs that are in phase 3 and the promise of, as Jordan Feld says, Perfectivir in 2020, industrial companies have us convinced that the problem of HCV is finished. But unfortunately, I think that there are a couple of considerations that still need to be addressed, and primarily that's price. The drugs currently cost about 50,000 euro. By early 2014, the promise is over 70,000 euro. I'm not a health economist, but it's quite easy to calculate that if you multiply that times 150 million patients, you come to 10.5 trillion euro to cure HCV worldwide, as compared to another important figure, which is the amount of global health expenditures annually, which is less than half of that. So basically, if we spent all of our healthcare euro on HCV, it would still take 2.5 years to cure. And obviously, neither this nor this are realistic numbers. And really, I think, is going to translate into HCV ultimately becoming a poverty-related disease, much more so than it even is today. And for this reason, I think that there's still a very important role for biomarker research. As I mentioned, we're very heavily invested in Egypt. It's a quite special and sad situation. It's the largest known iatrogenic epidemic in the 1960s and 1970s as part of a World Health Organization campaign to eradicate schistosomiasis. They traded one liver disease for another. 35% of the population became infected with hepatitis C genotype 4, and today it's close to 15% that remain infected. The annual budget that's available is about 100 million euro, a third of that coming from the Ministry of Health, and I'll come back to what that means in terms of the actual cure rate of HCV. So for hepatologists, the use of biomarkers is quite daily, in fact. We use them to treat to identify the infection. We use them to make decisions about who to treat. We have some very interesting biomarkers that are giving us information about the predictive effect of the treatment, as well as surrogate endpoints that allow us to withdraw patients who we do not believe are going to respond. In addition, there are toxicity biomarkers, and I'll highlight one example where we believe biomarkers has helped us understand disease pathogenesis. But the topic really is personalized medicine and personalized biomarkers. And for HCV, this is where most of the community is thinking, and we heard several very prescient talks this morning highlighting the challenge with utilizing the differences among us, between us, at the genetic level as biomarker information. And in fact, it was made already very clear that these single nucleotide polymorphisms are in fact not biomarkers. And I just want to drive that home. This represents the genome-wide association studies that have been run over the last 15 years, offering tremendous insight into the mechanisms of disease and translating into absolutely no new clinical tools that can be applied to the management of patients. HCV actually has the most highly significant, highest odds ratio GWA hit, which is IL-28B. I'm sure many of you have heard of it. This came to light in 2009 with several high-profile papers, odds ratios ranging between 6 and 24, depending on the patient population, astronomical p-values, certainly as far as an immunologist is concerned, and has led me to learn a little bit more about statistical methods in genetics. And I think that most of you are aware about the question of multiple analyte testing, the need for high numbers of patients to be able to perform some of these genome-wide association studies, and the importance of using correction factors that allow us to be more confident in our results. But again, these are questions of probability. I want to highlight as well that we need to concern ourselves with the prevalence of the SNP in the population, as well as the prevalence of the phenotype in order to translate into clinical practice. And I'm spending so much time on this because, in fact, IL-28B is used in many instances to try and identify those patients who respond to HCV therapy. And in fact, while not a standard of care, it is one of the few genetic biomarkers that have been utilized. So this world map highlights the prevalence of the different SNPs, and I'll focus primarily on the Duke SNP 860, where the CC allele, predominant in Asian population, makes them quite resistant to both spontaneous infection as well as susceptible to responding positively to therapy. And that's in contrast to African populations, which are more at risk to developing chronic infection and are refractory to pegylated interferon-based therapies. Unfortunately, when you start translating this into potential clinical tools, you'll see that allele frequency in African population of 0.3 translates into only 9% of the population being of the good genotype, where you would want to necessarily use this information to treat patients. And in Asian population, the 95% allele frequency into 90% of individuals being the CC haplotype. In both cases, I would argue that is not particularly useful. You want your biomarkers to cover more than 10% of your population. In Europe, we are a favorable split between the C and T allele, and in fact, something that is identifying correctly 40% of individuals that respond to therapy could be potentially useful, but as I already highlighted, the therapy has already changed. In addition to considering the criteria that impact the interpretation, we also have to consider the criteria that impact the utility of the diagnostic test: accuracy, precision, predictive value, the clinical question, it must be good, pretest probability. And I just want to highlight what one of our colleagues in Spain is doing, where, in fact, due to limited resources, they are trying to utilize IL-28B polymorphisms. This is the pretest probability, so no biomarker treatment. It's about 50% of individuals who are responding to pegylated interferon ribavirin therapy. introduced the IL-28B polymorphism, and now we see that we can identify correctly 75% of individuals who are going to respond to therapy. Unfortunately, if you, again, do the math, and this isn't very complicated mathematics, no biomarker testing, it translates into 45% cure rate. Biomarker testing, the overall cure rate is 26% of individuals because you're treating less individuals. And obviously, this is not going to be satisfactory to either the patient or the physician. And as a result, in most instances, physicians do not utilize IL-28B polymorphism. What I'd like to introduce is a concept, and I'll come very quickly to the application of these assays on point-of-care devices, but I think the concept is important, is that we can start utilizing intermediate phenotypes, something that is in between the genetic polymorphism and the disease, to be able to enhance the information that we currently have and perhaps begin developing clinical tools that will influence patient management. And by intermediate phenotypes, I mean RNA expression and protein expression that offer some functional information as to where that individual is in their disease pathogenesis and how environmental factors are influencing that individual's genetic predisposition. So again, in order to advance the promise of personalized medicine, we require functional biomarkers. And for us, that has come in the state of protein biomarkers present in plasma, something that is quite easy to access in regular medical care. And we have primarily been utilizing the Luminex technology, which permits multi-analyte profiling. And in collaboration with a company called Rules-Based Medicine, we now have access to over 250 assays in a CLIA-certified or ISO 15189 platform. Over the last 5 years, we have defined inflammatory signatures for different acute viral hepatitis infections. We've identified 5 analytes that distinguish spontaneous clearance and persistence, 11 that identify and distinguish chronic individuals who are ultimately going to fail or respond to pegylated interferon therapy, and 6 analytes that are recently identified that are distinguishing responders and non-responders to the new triple therapy, including the direct antivirals. I'm just going to highlight one, which is interferon-induced protein number 10, and bear with me for the next 2 slides. There's a little bit of immunology here, but I'll try and keep the jargon to a minimum. This is a chemokine. It is higher in patients who go on to fail HCV therapy. And what's interesting is that it was somewhat counterintuitive to be identified as a biomarker for treatment failure because this molecule attracts T cells and natural killer cells into the liver. And for that reason, we understood that it was going to help the clearance of virus. Relatively long story, very short. What we've uncovered in the past few years is that, in fact, there's an enzyme called dipeptidopeptidase 4 that is overexpressed in states of chronic inflammation, such as chronic HCV infection, that converts this molecule from an agonist into an antagonist that perturbs T cell trafficking. And in fact, in addition to it being an important biomarker, it's taught us a lot about new mechanisms of immune regulation. But back to the programmed presentation regarding biomarkers. In a prospective study, we've been able to validate the genetic polymorphism, IL-28B, a very good positive predictive value, not very sufficient negative predictive value. I've already highlighted the complication with utilizing this in the European population. Our IP10 protein biomarker, which has a reasonable positive and negative predictive value. But what's quite exciting is that when utilized in combination, the fact that IP10 is a linear variable, we're able to find a cut value that achieves a 100% negative predictive value, thus helping to identify those patients that really do need alternative therapies. And this can be tuned depending on what your specific medical question is. This has been validated in more than 6 independent cohorts, and those are listed here. We've worked with Myriad Rules-Based Medicine to commercialize the IP10 3-plex, and IL-28B is now available in several different commercial laboratories. And we've been quite excited about the possibility of utilizing this for patient management. Unfortunately, that has not been realized, and we've been trying to scratch our head and understand why that is the case. And I just want to highlight that I think this is a really unfortunate missed opportunity First, for the Eurozone, because I think that we could have made a difference. And I just highlight one example, again, sort of back-of-the-envelope calculations. We can assume 100,000 patients being treated. The prior standard of care was costing us $1.5 billion. The new treatments, it's now $5 billion to treat those same individuals. But that translates into 20,000 new cures. So that's great for the patient, not so great for the payers. Biomarker-guided therapy, the IL-28BCC, low IP10, maybe in combination with some of the other biomarkers we've identified along the way. We believe we've achieved a 95% positive predictive value and a specificity of 80%. As a result, we can identify of these 100,000, 40,000 that could have gone on the previous standard of care, costing $600 million to achieve the 38,000 cures. The others would be redirected to the triple therapy, and in total we would have saved 1.3 billion euro. It's even more pressing for situations such as Egypt and other resource-limited countries. There the budgets are quite fixed. There's no access to these new drugs. I mentioned already that we have $30 million US dollars available to treat HCV at the National Health Treatment Centers within Cairo. And currently, we're utilizing, without biomarker testing, pegylated interferon ribavirin. We get at a significantly reduced cost. We're able to treat 30,000 patients. That translates into about 15,000 cure. What we hope to be able to implement in the coming years is biomarker-based and biomarker-guided treatment, where we can improve this number from 15,000 to 27,000, increasing the cure rate by 80%, with the same budget, and directing the individuals who need it to other, probably still more expensive treatments. In considering moving some of these assays into Cairo, we bumped into another problem, and this is really where the point-of-care technologies has come in. And we've already heard a very impressive talk that has highlighted some of the reasons why point-of-care instrumentation is needed. The prohibitive cost of diagnostic tests, the lack of skilled workers in many rural sites, problem of reagent transport, chain of custody, stability issues are all reasons to try and move in this direction and enable personalized medicine. And we've just recently received finance for a new FP7 grant called Point of Care Testing for HCV diagnostics. And we are federating 3 different small-medium enterprises that have developed technologies that I believe are ready for field sites. Protein biomarkers that are read out on a battery-operated, essentially, Sony Discman, and I'll highlight in just a second the CD arrays that are being used for microfluidic-based immunoassays. The Epistem Gene Drive, which is a handheld PCR machine that is able to do very rapid SNP genotyping, and CluCore algorithms, where this company is going to fit our algorithms on IAPS that can be utilized by treating physicians. We believe that we'll be able to offer, for relatively low cost, diagnostic and theranostic tools that will improve the management of care in resource-limited countries. And just very quickly, the protein immunoassays, as I mentioned, are being run off of a spinning disk, taking advantage of the differential centrifugal force from the inside to the outside of the disk, moving the liquid through a series of channels and using plasma resonance-based technology to be able to quantitate the protein biomarker in 18 minutes. In addition, they've also developed systems that allow you to count the number of platelets, which may turn out to be a useful marker for adverse effects of treatment. And then the gene drive that is developed by Epistem is a rapid cycling heating block that utilizes the melting temperature of the product to determine the different haplotype of the individual. And in 45 minutes, we are proposing to generate information about IL-28B polymorphisms. They've also developed viral genotype testing and are working to develop viral load testing. This is just showing you some of the preliminary data where we're distinguishing the CC and the TT alleles and quantitative information. It is now possible to do this from a buccal swab. and then the full array of kits that are being developed by Epistem for those of you who are interested and engaged in resource-limited settings. It's quite exciting to see the way in which they're really optimizing different tests for infectious disease in southern countries. So ultimately, and in conclusion, what we want to be able to achieve is the integration of epidemiologic information, clinical status, genetic factors, immunologic factors, dose type of medication, as well as the health economic resources that are available for that particular community. And I think there are incredible opportunities for mobile unit surveillance, evaluation of side effects, as well as investigations in other disease. And I just want to end by 2 slides, maybe a bit provocative, but I rarely have an opportunity to speak to this community, and I think that there are many challenges where we need support. For point-of-care biomarker development, we need easier access to patient samples for biomarker qualification. We need support for multiplex biomarker testing, even more challenging across different technical platforms. And in fact, I'm becoming more aware of the challenges of the regulatory aspects of bringing biomarkers from 2 different arenas into the same algorithm. And then third, we really need partnering with physicians so they feel included in this process. The real tragedy here would be that we develop some algorithms, all of the tools to be able to help support clinical management, but physicians feel like they've been excluded. We don't and we can't tell them, obviously, what to do, and therefore it has to be a partnership from the beginning so that when these tests are available, they will be implemented and utilized. And then finally, maybe some action items that we could work on collectively, lobbying the EMEA and the French associations to insist upon companion biomarkers. It's quite horrifying to me in the current hepatology community that there's so much data and samples available. Many of us at academic hospitals have participated in those clinical trials. They're being utilized to justify the approval of new medications, but without access to those samples, without regulatory agencies insisting that they come forward with companion biomarkers, it's going to be the payers, in the case of France, the government, that is footing the bill for what are very expensive new medications. And I think that we could be utilizing them much more effectively by insisting upon cost-effective, personalized strategies. Second, the prioritization in the upcoming European Union grant applications for funding available for biomarker qualification and implementation, specifically focusing on countries in resource-limited environments. And it's unfortunate to see that some of those resources, at least at the European level, may not be available. And then third, establish guidelines for academic industrial partners that are going to permit academic partners to utilize those clinical samples that we have helped to collect. And I think that we really do need support from government in clawing back and ensuring that those are, in fact, equitable relationships between academic and industrial partners. I mentioned some of the people along the way, Stanislas Paul and Arnaud Fontenay and their teams. I'd also like to thank Dara Duffy, who's helped with the different European projects, and notably the formulation of the POC-HCV project, and Estelle Motet for helping us to collect and access some of those samples and the team that she's leading. And I thank you for your attention. I look forward to your questions.
Merci beaucoup. Donc, sans plus tarder, je passe la parole au dernier intervenant, donc Rodolphe Thibault, pardon. De l'ISPED à Bordeaux et de l'INSERM.
Merci beaucoup pour cette invitation, Jean-Paul, les experts. Merci aussi beaucoup à l'équipe organisatrice. C'est absolument parfait. Je suis enchanté de présenter, même si après toutes ces présentations brillantes, je vais devoir vous parler un petit peu de mathématiques en plein milieu de l'après-midi. C'est un challenge, mais en fait, et du coup j'en profite pour vous donner le take-home message immédiatement si j'en perds quelques-uns sur la route, c'est qu'on a beaucoup parlé de tous ces big data, on parle de ces implications en termes de personalized medicine. Il y a un souci, il faut qu'on les analyse ces données, et pour ça, on a besoin de plusieurs métiers, des méthodologistes, des épidémiologistes, des bioinformaticiens et des biostatisticiens. Et c'est un tout petit peu de ça que je vais vous évoquer maintenant. Donc je travaille à Bordeaux, dans le centre épidémiologie et biostatistique INSERM 897, et dans laquelle je dirige aussi une équipe INERIA système. Alors, donc on va revenir un petit peu sur l'aspect personalized medicine, et j'évoquerai seulement certains aspects pour Nord-Sud et sur le cost effectiveness, mais en tout cas, ici, on va parler un petit peu d'innovation. Mais avant, je crois qu'un des points qui est vraiment majeur pour la problématique de personalized ou stratified analysis, on a besoin de bonnes données. Alors pourquoi je mets entre guillemets bonnes? Parce qu'on a vu, il y a tout un tas de critères de reproductibilité, etc., dont on a besoin. Et puis il y a un autre aspect, c'est que ce matin on a beaucoup parlé de medical genomics, de données génétiques, etc. Mais la personalized medicine, on a revu ça à travers d'autres exposés, ce n'est pas uniquement potentiellement les aspects génétiques, génomiques, bien qu'ils nous apportent énormément, les données omiques nous apportent énormément d'informations, mais il y a aussi toute autre information qui peut être pertinente selon le contexte dans lequel on se situe. Et puis après, il y a donc les méthodes qu'on met en œuvre, les méthodes statistiques. Et là, c'est pareil, il y en a énormément. Alors bien sûr, j'aurais pu faire un truc horrible qui est de faire un catalogue de toutes ces méthodes qu'on peut utiliser en le liant avec personalized medicine, ce que je ne ferai pas. Simplement évoquer que voilà, il y a des méthodes très autour de l'apprentissage, du machine learning, et par exemple pour faire le lien justement avec le Nord-Sud, il y a un papier qui vient de sortir dans le dernier Journal of Antimicrobial Therapy où il y a un modèle prédictif basé sur des forêts aléatoires montrant qu'ils arrivent à prédire totalement, enfin aussi bien que des tests génotypiques sans utiliser des tests génotypiques dans le contexte VIH, avec bien sûr les implications que ça peut avoir dans le Sud. Il y a d'autres approches, on va y revenir, mais ici en fait ce que je vais faire,